Welcome to the GDC June 2022 webinar. My name is Zhenyu. I'm co-PI of the GDC and also the bioinformatics lead. In our previous webinars, we have talked about some of the more commonly available data types. This includes RNA-seq analysis, like whole exome variant calling from ECF to MAPS. In today's webinar, we are going to talk about some data types that's it's not very commonly available across all the projects. And also, we're going to talk about some of the data types that's probably hidden a little bit deeper, and you need some effort to find them. It's not very well known to most of the users. In particular, we are talking about the 5 data types here, and at the end, the later part of the seminar, Bill will give a live demo describing how to access this data type. Okay, so in today's webinar, I'm going to first talk about whole genome variant calling. This will be followed by 3 array data types, including a classic SNP6 genotype array, methylation array, and a protein array. And the last topic I'm going to cover is the analysis results GDC have derived, but not served as a file. They are served as BAM properties available through APIs or web portal. Okay, so let's talk about the first data type, whole genome variant calling. For the whole genome variant calling, in particular, we're talking about tumor normal paired somatic variant callings. The GDC has adopted a set of workflows from the Wellcome Trust Sanger Institute, in particular their Cancer Genome Project. This set of workflow contain actually 4 different tools. 2 of those are for short mutation calling. This includes Caveman, it's a particular point mutation caller, and Pindale for indel calling. We also have a somatic copy number variant caller called ASCAT-NGS. It uses an algorithm called ASCAT. As I mentioned, in a little bit later part of this presentation, I'm going to talk about a genotyping array, a SNP6 array. It will also be analyzed by a similar algorithm, and you'll see a workflow called ASCAT2. It will generate exactly the same kind of output. So I will move this discussion of the ASCAT-NGS into that portion of the presentation. And I'm going to cover the 4th caller here. It's a structure variant caller called BRASS. In this particular BRASS output, there are 2 types of files. One is VCF file, the other is BATP files. They are roughly equivalent. By equivalent, I mean if we have a variant record in the VCF, it will have a corresponding record in the BATP. The reason of having 2 different formats for the same information is because, as we know, the VCF 4.0 is not the best format to store a structure variant information. And also VCF is a little bit not very as convenient as the BATP or as like tabulated format for users to use. And BATP is something we expect most users will actually download and look into them. So what is BATP format? If I assume most of people are very familiar with the BAT format, which describe a genomic location in terms of the start and the chromosome and strand. BATP is a natural extension of the BAT format. It's, as we know, for structure variant, we have a secondary endpoint. So it provides, in addition to description of the first breaking point is describe a second breaking point by providing the chromosome 2, start 2, and end 2 information, or end strand 2. In this format, the first 10 columns are required. So in addition to this column I've described, there are also names and scores. Although names, scores, although they are required, the exact definition of those are not defined by the battery format itself. So it depends on each individual caller how they put a name or put a score for them. After these 10 columns, there are all the optional fields anyone can write down. And if you download look at the BRAS BAT-P file, you'll see a lot of different columns. And they provide very important color informations, annotation informations, or even categorization of different structural variants in this file. And if you really interested in how to use this data format, you can go to this link. This is a direct link to the Cancer Genomics Project Center, and they have a more detailed instruction on how to read this particular BAT-P format file for BRAS. So this is the current status of the whole genome variant calling. And if you're familiar with the whole exome data available in the GDC, you will know that for whole exome, not only we have the raw VCF called from each individual callers, we will add additional filtering workflows and annotation workflows. And this will be followed by VCF to math workflow, and finally we'll have a protected math that contains most of the information, and the public math that's being extensively filtered, including some of the strategy like we require the variant be called by at least 2 callers. So it's the final mask ensemble math is the one have been of the best quality and what we expect users to use. In terms of the whole genome workflow, the current status is we have implemented the raw calling step. So you can see some of the whole genome VCFs available in the portal. The additional annotation, filtering, and especially the math ensemble workflows are not available yet. We are actively working to get them implemented. In particular, if you look at the workflow, you can see we only have 2 small mutation callers. One is point mutation, one is indel. So if we require the 2 caller ensemble, probably we don't have anything left. So we are actually also adding additional small mutation variant callings for the whole genome data. After this is done, and we expect the workflow on the current schedule to be complete by the end of this summer, we'll roll out a production phase and gradually to release this whole genome maps in the portal. And I think we should expect those whole genome maps to be available from the beginning of next year through different projects, probably one by one, because they need a lot of computation. But our purpose is eventually all the data we have whole genome variant calling will have similar filtering, annotation structure, and similar maps as what we have for the whole exome workflow. So that give us more consistent and more easier to use data types to do the whole genome variant analysis. Okay, so I'll move to the first of the 3 array data types I'm going to cover today. This is a classic Affymetrix SNP6 array. I think Affymetrix was later bought by Thermo, so it's a Thermo SNP6 array now. This is a traditional array type being used by TCGA. And in the GDC, two types of analysis are available. On top is we call the old workflows. They're shown in green. And the bottom is the new workflows that basically what we just mentioned, the ASCII2 workflow, shown in yellow. The old workflow, we use a very similar method as what TCGA original HG19 analysis have been used. Actually, we use one of the intermediate file that analysis have generated. That's the probe intensity file. And instead of generate the HG19, we just redo the circular binary segmentation using DNA copy package and generate the data for the GDC portal as GRCH38. If you have been using those segmentation files a lot, you'll be very familiar with what's the format. So I'm not going to cover it here. Just one notification if people not familiar with the segment mean value being provided. The segment mean value normally is defined as the log2 scale of the absolute copy number, subject 1. What that means is if it's a diploid locus and we got the absolute copy number being 2, so this segment mean is 0. For anything that's less than diploid, have a copy number loss, you expect a copy number segment mean negative value. If you see a positive value, that means a copy number gain. And there are also differences between the segment copy number segment and the mask copy number segment. In this mask copy number segment files, some of the low probes that have been more easy to be variable in the germline settings being removed. This is consistent with how TCGA did that previously. So the mask copy number segment is what we expect most of the users will take for the arithmetic analysis. Okay, we also mentioned GDC has a new workflow using ASCII2. It generates very different output. It has a copy number segment, but you can see it's called allele specific copy number segment. I will describe why it's allele specific. And also we generate a gene level copy number. Okay, so if you open this allele specific copy number segment file, you'll see very similar first level column, but then you have, instead of a copy number segment mean, we have a copy number value that being an integer value, and that's the exact copy number. When I say exact copy number, that means if the value is 2, that means diploid. 1 means 1 copy deletion. 0 means the entire region is lost. And anything above 2, that means the exact copy number of this locus. And this use a algorithm called the ASCII. You can click here. The news lab got the exact algorithm. It does something similar as what Rhodes have the absolute analysis. If you have used that, it's trying to estimate the purity and the ploidy at the same time, and then use the purity information to derive for each segment what kind of copy number values we have. And in addition to this one particular value, it also provides a major copy number and a minor copy number. That's why it's called allele specific. Basically, the major copy number and the minor copy number, their sum is this total copy number, and the major copy number is defined as always equal or larger than the minor copy number. So not only have the total copy number, you also know how from the original two strand how they now distribute. Sorry, and original two chromosomes how they distribute. Okay, and also mentioned there is a gene level copy number generated out of this algorithm. It's as simple as we overlay the gene regions to the segment regions and check what kind of copy number this gene region inherits from the segment. There are a few caveats. I will describe in this diagram, but basically we have a copy number, and if a gene have overlapped with multiple copy segments, the smallest value of those segments will have been written here as the minimum copy number. The max value of this copy number segments will be written here as the max copy number. In most of cases, they are the same, but sometimes for this example, you can see the max is really larger than the minimum. So how this copy number got inherited from the segments, you can see in this diagram. I have labeled 4 genes, and there are 4 segments also derived from the ASCAD algorithm. So for the first gene, it doesn't overlap with anything. So we don't know its status. We just keep it blank. That's the first row. The second gene, it overlaps with segment 1 completely, and it inherits the copy number as 2. The third gene overlaps with 2 segments. Both segments have a copy number of 3. So this gene has a copy number of 3. The fourth gene is the most complicated one. It overlaps with 2 segments, and these 2 segments have different copy numbers. So what we do is we record the minimum copy number as 3, the maximum copy number 5. So how we do the copy number? This copy number is a weighted mean of this overlapped region. So in this region, this gene overlaps with segment 4 more than segment 3. So as the weighted median, not the mean, weighted median of the copy number value, we take 5 as its copy number value. So you can see for this particular gene, the copy number is labeled as 5, minimum as 3, maximum 5. So actually, if you see any genes have these 3 values not consistent, that means more than 1 copy number segment have been involved here. If you are really interested in a more detailed analysis, you probably want to really look at the segment themselves and see how they will affect the gene. Sometimes maybe just a non-transcribed some other or the 5' end being modified instead of the main gene body. But that's something you probably want to pay attention to. Okay, the second array type I'm going to talk about is the methylation array. Methylation array is also a TCGA classic array type. In TCGA, there are 2 subtypes. One is human methylation 27, and the other is 450, which have 27,000 probes and 450,000 probes. Both are chips from Illumina. And later, Illumina have a new chip called Epic Array that have 850,000 arrays. We have used a completely new workflow compared with what TCG have used. This workflow is actually derived by the same lab that's analyzed the classic legacy TCGA data, and they have a lot better probe QC and a lot better accuracy in terms of the output. So this is definitely more superior than the legacy TCGA methylation analysis. You can read this paper. It will also describe how they did it differently and why it's much better. And the output data looks like that. I only have two columns. There's no file header because some program may want to take the file directly. Right? We don't want to add additional non-standard headers. But the first column is the probe ID. The second column is the beta values. If you want to look deeper into this and see how they interact with the other data types, you probably want to know where is this probe, which gene it affects, and what kind of CPG island it's located in, or as a neighbor. So I really encourage you to dig out a reference file from the GDC reference file page. If you haven't used this page, please go looking for it, called the GDC reference file page. We have all the FASTA file, the gene models, the star index file, but also we have things like that. This is the methylation array metadata information. It from every probe, you can see the probe ID that's matching with what we have in the data file itself, but it also will tell you where is this probe and which gene this probe affected, which transcript this probe affect. What's the distance from this probe to the TSS of each gene? And what are the CPG positions around CPG island or CPG shores? And what exact type of the CPG is an island, shelf, shore, etc., etc. You can get those definitions from the paper I described above, or go to Illumina website and find their description. That's give you a lot more information, especially if you want to do multi-omics analysis and checking how this methylation CPG cluster affect the gene expression nearby. Okay. And the third of the array types I'm going to talk about is called reverse phase protein array. This is an analysis being performed by the MD Anderson Functional Genomics Core. And I think we have such data in TCGA and CPTAX3 as well. So what is this reverse phase protein array? So we know for most of the array we talk about, we probe like, we get the probes or or any like antibodies to the slides, and then we flush the slides with samples, solution with samples, and we detect signals. This is the opposite way. Actually, in this kind of array, different samples are actually plotted onto the slide, and the slides will be flushed by solutions of antibodies. So that's why I call it reverse phase, because it's done differently. It does also have controls, but the event purpose of this is to derive expression of proteins or modified proteins, for example, like sulfated protein in the sample. And the analysis and normalization, everything have been done and it's completely done in the MD Anderson using their pipeline. So GDC is not involved in the complete analysis. And in the output file here, I provided an example. What you can see is basically it gives you, for each sample, give you in this sample what's the peptide target. Like, for example, like you histone modified H2B, this give you a normalized expression of -0.6. And for some of the other protein, like twist, give you a normalized expression. Those normalized expression can be used to do comparison across different samples. This file also contains some other information about this particular antibody or the particular antigen it's trying to detect. So it have an antigen ID, lab ID is a antibody ID. Catalog number is the catalog of this vendor, and set ID is a little bit interesting. So the MD Anderson Center often have like I query 173 antibodies in this analysis for a period of several years. And now I have validated 15 more antibodies. I added these 15 more antibodies to my old set. So I am querying 220 antibodies. So they will create a different set ID. It's kind of a little bit related to the batch. So that's how they have this set ID. So this is the file you can download it from the GDC portal. Similar to the methylation array, we also have a reference file that provides a little bit more information. And in addition to what we have described before, it also mapping each antigen ID with the particular gene that's the gene code 36 genes GDC are using uniformly across the portal. And it will tell you the origin of the antibody. For example, it's a rabbit antibody. For the presentation purpose, I remove the source. The source basically is the company. You can go and buy this antibody. And you have a category number as well, so it makes it very easy. And MD Anderson also have used antibody. Sometimes later they find, well, it may have some QC issues, so they will label whether this antibody is valid or invalid, or they haven't in time to looking at the QC, or it's a little bit vague. So this is a antibody quality validation status. You probably want to look at it and only pick the ones that's validated. This file is also available from the GDC reference page. Okay, so I've covered all the three arrays, and finally I'm going to talk about GDC analysis and BAM properties. And a lot of people, most of the people probably doesn't know that GDC have analysis results that are not saved as files. Why that's the case? Because some of the analysis results are just one value, and having those as files are redundant for people to use. And also, we do want to use the majority of this analysis result as a facet filtering property in the GDC portal. So they will actually present it as GDC graph node properties. And in particular, the BAM node, or in GDC we call the aligned read, it contains the most useful properties. You can go to the GDC dictionary viewers, just look at the analysis reads and go through them, looking at all the properties. You can have a good idea how many different useful properties we have that may be valuable for you. How we can get this data? There are two ways. One is we can, through the API call, and back the property value directly. And the second, we can also add them as custom file filter in the GDC repository for facet filtering. I will describe them in my slides, and Bill will also talk about them in some of the demos. So the first analysis property I'm going to talk about is microsatellite instability. We know this is a very important analysis for colorectal cancer as well as some of the other minor cancer types. We use a pipeline called MSI sensor 2. The MS sensor is a pipeline developed by WashU, and this is a updated version of that. And essentially, this workflow allow this analysis to be done from tumor data only without the normal pairs. It derived two values. One is a percentage of the site contained MS instability, and this gives us a value between 0 to 1. We call it MSI score. And then there's a cutoff applied, and currently this cutoff is called 20%. So anything above 20%, GDC will call it MSI, so microsatellite unstable. Anything below, we will call it microsatellite stable, or MSS. Here I give a example of how we can use the GDC API to get this data. So if you're familiar with the GDC API, or if you're not familiar with, you probably want to read the GDC API documents. We have the API standard API call. You can just copy and paste that in your browser, or you can use curl to get them. So it's the API call, and we're calling a file API, and we put the BAM file UID here. This is the BAM file UID. We have some formatting parameters, and the API is asking for the following 4 values: the MSI score, the status, the file ID, and file name. Just put it in the browser. It will give you this information. This file is this file. This is exactly the same one as we are asking for. And it will tell you the MSI score is about 5.7% because it's lower than 20%. So this data is MSS. Just one thing I need to add is this is the GDC analysis result. And we are not a clear lab, so this is saved as a BAM property, means a molecular property. In some of the projects GDC accept, they do have MSI analysis, especially for some of the clinical trials involving colorectal cancer, this being one of the biomarkers. This project may have used some clear labs to derive more accurate microsatellite analysis result, and this MSI status may be saved in some of the GDC clinical data file instead of these BAM properties. So if you go into the GDC viewer, you can see some of the clinical molecular analysis, molecular tests. They have another microsatellite instability values. Those belong to where normally clear lab have derived and submitter provide us. I would expect most of those are similar to what we have derived, but pay attention because what we derived are molecular tests, and the same file may have, same patient may have different files, and we derive multiple different MSI status depending on each file that being submitted to us. And sometimes there will be very rare cases you see inconsistency between those files, or inconsistency between the file we have derived from what the KLab have derived. It could come from different reasons. One common reason is we probably use different samples. Another reason is maybe this is a borderline MSI status that happens. So what I suggest if you find a party that already have a clear lab submitted uh, clinical data, have MSS sense data, you probably want to use that one instead of the molecular derived one. Okay, and I also mentioned this can be very used as the customer filter, and Bill will describe it later in the live demo. Other BAM properties, I will take them together. You can do a similar query, but in addition to what I have used for MSI, you can ask for tumor purity, tumor ploidy. These are the two properties being derived by the whole genome analysis. And you can get total number of reads or even coverage. This being derived by SAM tools or Picard tools. If you're looking for contamination, that's one of the GATK tool give you a percentage of this sample being contaminated by a sample from another patient. Normally, in a GDC perspective, if we saw a BAM file being more than 4% contaminated, we'll try not to derive or release variants from this BAM file, because a lot of variant derived will be actually the germline variant from its contaminated patient instead of a somatic variant from the patient that we are interested in. And there are a lot more that you probably want to go to the dictionary viewer to look at them, but if we just do the query I just mentioned, you will get a JSON payload give you the coverage of this whole genome file is 65x, 666x, and the ploidy is almost deployed, and purity is 63%, pretty pure, and also have contamination with very, very low, so it's a very good quality BAM file. Okay, that's all I have. I will hand this to Bill.
Thank you, Zenu. I'll go ahead and share my screen. Sharon, could you allow sharing for me? Doesn't seem to be working right now.
Sure, let me go ahead and do that.
Just a reminder, if anyone has any questions, feel free to type them in the Q&A field. I don't see any right now, so I do have time to give this demo, but if you have any questions while I'm giving the demo, we'll answer them at the end.
Okay, Bill, you should have it.
All right, thanks, Sharon. So I'm going to walk you through a few points on how to access the data that Jenny just talked about. So in order to access most files, you would go to the repository field at the portal, which is at portal.gdc.cancer.gov. So if you want to look up specific file types, you'll want to be on the left side in the File tab. And a good place to start looking is the Experimental Strategy field. The first file type that Jenny talked about was whole genome sequencing. Very easy. You just click on whole genome sequencing, and among the data types, you can see all of the things that he talked about, such as the structural rearrangement, copy number segment, gene level copy number segment, etc., as well as the aligned reads, the raw aligned reads. And each of the workflow type is also aligned here. So these typically correspond with the data types, so either one can be used to filter. Next is the copy number data, the ASCAT data. Now, in terms of experimental strategy, this can be found under the genotyping array. If you can't remember that directly, you can access them using the different workflow types like DNA copy and ASCAT2, or using the platform. A lot of people are familiar with that, the Affymetrix SNP6. So that's how those files can be accessed. As you can see, we have the different copy number segment files and the gene level copy number, as well as the segment from DNA copy and the mass copy number segment, which has some of the regions stripped out. Next, the protein expression data. that can be accessed in by selecting the reverse phase protein array data. And when that narrows things down, you'll see that the data type is also called protein expression quantification. So that's how these can be accessed. These ones don't actually have a workflow type because these are files that were submitted directly to the GDC, so no actual GDC workflow was used. So don't count on that field to query these files. And lastly, as you might have guessed, the methylation data can be accessed using experimental strategy. Just click on methylation array, and the Sesame methylation beta estimation can be used to access the masked intensities, which remove some of the germline specific areas, and the methylation beta value files that Zenu went over in his presentation. Now that you know how to filter for these specific files, I want to show you how to filter for some of the properties that Zenu talked about before. So in this case, you'll want to be in the Files tab. Most of this will take place in the Files tab. You'll click on Add a File Filter, and while these filters all down here are the ones that are default, Add a File Filter will allow you to choose from the many other filters that the GDC has to offer. So in this case, there's two that I want to focus on. Those are the MSI score and the MSI status. MSI score. And you'll see there's a few of them here. You want to click on the MSI score plain by itself. So you'll click on that. And then the other one you can add, MSI status. And if you're wondering, the difference between analysis input files, MSI status, and MSI status is that MSI status is directly applicable to the BAM file. So if you filter by MSI status, you'll get the BAMs with the specific status, the alignments. If you filter by analysis.input files MSI status, you'll be searching for the files that come from the BAMs that have an MSI status. For example, VCFs and MAFs that come from a BAM with a specific MSI status. So we'll search the plain ones for now. And as you can see, you can query by MSS stable and unstable. So here are the stable ones here. And if you wanted to be a bit more precise, you wanted something besides the 20% cutoff, that could be arranged by typing in the score and your own custom range here. So that's how these filters are used. And this is unrelated, but just a common use case is that a very similar thing can be done in this Cases tab by clicking Add a Case Biospecimen Filter, and you could do that for something like sample type or tissue type, which isn't directly in the defaults. So the last thing I want to do is show you how to access all of the BAM-QC features for a specific group of BAMs. And this can't be done directly through the portal, so I'm going to show you how to do it via the API, and hopefully have some time for questions. So we're going to go ahead and filter us a list of BAM files. So let's go for the CPTAC2 project. Let's filter by whole exome sequencing. And then we only want to look at BAM files, so we'll narrow it down to the data format BAM. So now we have 668 files and across 340 cases. So I'm going to go over to my terminal here, and I'm going to show you how to use the API to find this. So I'm using curl. I know other people like to use other things such as a browser, but curl works fine for me. api.gdc.cancer.gov is the main URL, and I'm going to type in status here just to make sure we're looking good. I expect the API to work, but I wanted to make sure I was putting in the correct URL. So we're going to be querying the files endpoint, and we're going to put in some parameters here. The first of which is going to be the filters parameter. And what this is going to do is, by itself, the files endpoint just gives you all files by default. Filters will give you will we'll narrow it down to a specific group. So we're going to go back to Chrome, and a JSON file can be used to pass information directly to this. But what we're going to do is we're going to cheat and go directly to the portal, which builds this JSON for you. It's URL encoded. So I'm going to, after this filters equals parameter here, I'm going to copy everything here all the way to the end, and I'm going to paste it in the URL decoder encoder. And the reason I'm doing that is because some of these quotation marks don't work well in the terminal. So I'm going to go ahead and decode that and re-encode it real quick. So now it's very clean looking, and I can take that and I can paste it as my filter in the terminal. So I'll go ahead and paste that. And real quick, let's just make sure that works. And I think we're good. So a way to make this a bit more human readable is to type ampersand, which brings you to the next parameter, format equals TSV. And we're going to go ahead and pass this to the less program just so that we can look at it a little bit. This looks like a mess, but I'll straighten out these lines. And as you can see, you have this parsed out in a nice TSV format with all of the BAM QC things such as average base quality, average insert size, average read length, separated out into columns. And something like this could be saved to a file, and that can be opened in Excel or Google Sheets or any spreadsheet program you're using. So, and you'll see that there's only 10 here. I only decided to use 10 so it would go quicker. But what you could do is you could add another parameter onto here that says size equals, and then something bigger than the 600, and that'll give you everything. That might take a second. But that is the demo. And the last thing I want to show you real quick before we get to questions, I was hoping that was going to finish before I stopped talking, but there it is. Look at that. Anyways, the last thing I want to show you is the list of fields that can be queried for a BAM. So the way you can see this, and Zenu pointed it out in his presentation, is you would go to docs.gdc.cancer.gov, our documentation site. You go to the data dictionary tab, which I'm highlighting here, and you click on viewer. Once the data dictionary viewer loads, you'll see a list of different entity types, and what you want to find are the aligned reads. We have submitted aligned reads, but you want aligned reads. So you'll click on that, and as you can see, there are a list of all of the different BAM-QC fields that can be populated at the GDC. So if you are wondering what the GDC uses for BAM-QC, a list of them can be found here. Thank you so much. That is the end of this demo and this presentation. We will take questions now.
Okay, Bill, there's a couple of questions here in the Q&A. Are there other well-detailed tutorials out there to complement this in a step-by-step process to analyze any of the data?
So that is a good question. So if you have any questions on how to use the API and some use cases in the GDC, I'd recommend going to our webinar page, which can be found at gdc.cancer.gov in the support section. That has a good list of them. In addition to that, if you want to look a little bit further, there are plenty of GDC use cases on YouTube. These are not they are not controlled by us, so there's not much we can support in terms of them, but I know that many other users are giving their own demonstrations of the Genomic Data Commons, as well as BioStars. I would check out that website too. There's lots about the GDC there.
Thanks, Bill. There was a somewhat complimentary comment, a question in the chat that was made about demonstrating the entire analysis workflow in a practical hands-on format. You know, and I think that would kind of, I know we have other webinars that do support, that, but I didn't know if you have any additional details on that.
I don't have anything specific about that, but that could, if that could be a good idea for a future webinar, specifically about some other analysis that we haven't done yet.
Yeah. And for the existing analysis we have done in the GDC documentation, we have lots of information about how this workflow was constructed. Sometimes there's detail as the exact command line this workflow is built. Two of the workflow we mentioned here, one is the Sanger whole genome variant calling workflow. For this one, we adopt directly what Sanger have provided. So we have a link over there. It's easiest to just go to the Sanger documentation, and they provide a lot more information than what we have. And also the Sesame workflow, we have some description in the GDC documentation how this is working, but there is a more detailed 2018 paper published on this pipeline, the Sesame, and describe how to use this workflow. And that actually they also provide a very good use case how they actually use the data derived from this workflow to have identified something interesting that you wouldn't find out with the older analysis and only be available with the new Sesame analysis. So that's something you probably want to look at as well.
Thanks. And the next question is, will whole exome, I'm sorry, there's a pop-up that's blocking my question. See if I can get to it.
I can read it.
Can you read it?
Will whole exome or whole genome based allele specific copy number estimations be available?
Yeah, I saw look at both questions. So the whole genome is what we have talked about. The whole exome, we currently don't have a plan to call ASCAT algorithm on the whole exome data. Whether we will do it in future, maybe we don't have a plan yet. But just to let you know, like what we have observing is in this recent years, whole genome gradually become the dominant file format. We have more and more whole genome data submitted to the GDC than whole exome year over year. And even for legacy project like TCGA, I think the NCI OCG is trying to sequence more whole genome of the same sample. So even the legacy project may be updated with more data, whole genome data. So stay tuned. We will probably release more data on the legacy project, and a lot of the new project will have a lot of whole genome data analysis.
Okay, are there any other questions? I think there's one in the chat. A question about accessing the controlled BAM or other file formats. Are they sample matched?
So
Go ahead.
so the control BAMs are they are sample matched in some cases, other ones are not. It depends on which project and which specific subset of data. This can be figured out by usually which of the BAMs have corresponding VCFs and MAFs. Usually, if they don't have variants, it means that they either haven't been called yet, or there's not a matched tumor or normal. Like it's only one or the other.
Are there any other questions? Okay, there's a question. Can ASCAT2 be applied to detect germline copy number variants?
That's a good question. I never thought about that. I think Based on my knowledge, it probably cannot, but you really need to go to the origin developer to know what they have and for that.
Okay. And is it possible to submit our own data, 40 whole genome samples to the GDC? Are there any territorial restrictions?
So people do submit their own data to the GDC. We have a process in which the GDC leadership will assess whether or not a subset of cancer data is a good fit for GDC. So I would recommend going to gdc.cancer.gov and clicking on the Submit Data field. There's lots of information there. And I do want to add that this requires that there is a manuscript associated with the data and that you have a dbGaP project associated with the data. There are some other restrictions too, but those are the main ones that users are typically interested in.
Okay. Any other questions? I think we have about 2 minutes. Okay, I do not see any more questions. Thanks everybody for attending the webinars. As indicated before, it's going to be made available on our webinar website page. And thank you, Bill and Zhenyu, for this wonderful presentation and demonstration. Everybody have a great rest of the day.
Thanks, everyone.
Thank you.